Hello, this is Mrs. Kasperwitz, and in this video I'm going to introduce you to our first kind of tissue in Chapter 2, which is epithelial tissue. So yesterday you did an activity where you were introduced to the 4 basic kinds of tissues in the human body and got a brief introduction to those tissues and what they do for us. So today we're going to focus on the epithelial tissue, where it is, its structure, and some of its many functions. So let's start first with where our epithelial tissue is. So the name gives you an idea of where and possibly a function of the epithelial tissue. And epithe, right, is a prefix that means to lay over or cover something. So our skin would be a perfect example of epithelial tissue. Our epidermis, which is the outermost layer of our skin, is made up of epithelial tissue. The skin is the largest organ in the human body. If you were to kind of take it off and spread it out flat, it would take up about 2 square meters. And it accounts for about 15% of your body weight. So a person who weighs 100 pounds, about 15% of their body weight, or 15 pounds, comes from their skin. So where do all those shedding off skin cells go? Well, they end up in your they're dust, essentially, or they're part of dust. They get in your bed, they get in your clothing, they land on the floor. And dust mites then use them as a food source, which is kind of nasty, and you will, vacuum them up and mop them up when you do those activities. So they really do become part of the dust that's in the environment. Dust. So that's the big one. So epidermis is a major kind of epithelial tissue. And the other place we find epithelial tissues is in the lining of tracts within the body. So a tract is simply like a tube, all right, that either goes all the way through the body, like the digestive system, all right, or has an opening to the outer environment. So for example, your digestive system, you have a mouth and you have an anus, and then basically there's just a long tube that runs all the way through the body, all right, that's the GI tract. And the lining of the GI tract is epithelial cells. And some of those epithelial cells are going to be absorbing materials from the environment, from the tube. Some of the cells in that tube are also going to be glandular in nature, where they're going to be secreting materials into the tube itself. So those epithelial cells are lining that tract. In your urinary tract, so again, there's an opening to the environment at the urethra. And then there's tubes that go inside your body, so the urethra, the ureters, up to the kidneys, and the bladders along the way there, right? So that all of that tract is lined with epithelial tissue. And we can continue this on and on, right? So reproductive tract, so in females, for example, your reproductive tract, so your vagina, your uterus, your fallopian tubes, all are lined with epithelial tissues. Okay? So very important part of the body. So here are some examples, right, of some epithelial tissues. We have, again, here's your epidermis. Those are the epithelial tissues that we're talking about, that we're shedding off a moment ago. Here we have a goblet cell. This we're looking at the respiratory epithelium. All right, so this is lining your respiratory tract, again, open to the environment. So we're going to have epithelial tissue running right down the mouth and the nose, into the pharynx, the trachea, and so on, down into the bronchial tubes and the bronchioles in your lungs. Okay, and I believe this is, I'm not exactly sure what this is. I want to say this is one of the tubes that are in the urinary system. But again, you can see the epithelial tissue here is in the lining of that location. All right. So the third kind is the glandular tissue. So not only does the epithelial tissue cover and line the tracts, but also when you have a gland, the glands are also modified epithelial tissue. And so these are glands that have a secretory function, so they're going to be making something that's going to be put out into a tract or a tube and moved some other place in the body. It could go into the blood as well. So these include both your endocrine glands that produce your hormones like insulin and glucagon, and they also could be the goblet cells, which we just saw in the previous picture, in the respiratory system. They could also be exocrine glands like sweat glands. So we'll be talking more about those later on this year. So here are some examples of those glandular type situations. So these things right here would be the glands, which are, again, modified epithelial tissues. And again, here's your infolding, all right, creating a gland situation there. So what are some functions? So the epithelial tissue is forming boundaries between different environments. So clearly your skin would be a boundary between the external and the internal environment of your body in general. That would be for protection purposes as well. In your linings, right, so the epithelial tissue is going to be what's in contact again with with the external environment. So that lining is forming a boundary between the tract, which is connected to the external environment, and the cells and tissues underneath there. Talked a little bit about absorption. So one of the cells that line the small intestine and the cells in the lungs that create the alveoli, those are for absorption purposes. You can have epithelial tissue that does some filtration. And then we have, again, the secretion part of that. So epithelial tissue that might do filtration would be in your kidneys, where we're going to be, taking some of the waste products out of the blood and then excreting them in the urine. So those are some places where you might find epithelial tissues and their functions. So what are some characteristics of these tissues in general? We talked in the beginning part of this unit about the desmosomes and the tight junctions. So tight junctions are where you've got proteins that are holding the cells together in impermeable sheets, right? So on the test we just took, the example of that was the blood-brain barrier. So between the blood vessels and the nervous tissue in the brain, we have this epithelial tissue that is forming this impermeable sheet of cells so that materials that are not good can't pass from the blood and into the nervous tissue. Tight junctions also hold together cells that are in the skin as well. We have desmosomes also there. Desmosomes are going to help with like mechanical things. So like we don't want, if you're rubbing on your skin, you don't want the skin cells to pull apart from each other. So there are what we call desmosomes, which are going to hold those cells and help them protect them from mechanical friction. Sorry about that, my camera keeps looking at me. The exception to that is the glandular epithelia. The glandular epithelia do not have desmosomes and tight junctions, so we don't necessarily want them to be impermeable sheets or sheets that need to deal with holding themselves together against mechanical pressures. Epithelial tissue cells tend to have what we call polarity. All right, so there's an apical surface that is facing the environment, right? So if we're in a tract, the free edge or the apical surface is going to be pointing toward the environment, and then that cell is going to be attached to what they call a basement membrane. So it's got another side to it. And so we'll take a look at one of those in just a moment after we finish the characteristics, and I have some pictures to show you. Some other interesting things that you might not know about the epithelial tissue is that it itself has no blood supply of its own. We will talk more about this for the skin in the next chapter, but it is avascular. That's the fancy name for no blood supply. So vascular tends to refer to blood vessels. If you're A something, you don't have them. All right, so there's no blood or capillaries or things going to that area. However, it is innervated, and that means that it is connected by nerves to the nervous system. So your skin, right, can have nerve endings in the epithelial layer, in the epidermis, but it won't have blood vessels flowing in that. That's why, like, those upper layers of skin you have here, like, you don't necessarily bleed when you pull some of those layers off. I know that sounds gross, but it's true, right? And that's because it's avascular. If you provide epithelial tissue with enough nutrients, they can regenerate themselves, and you know, they'll just keep going like little Energizer bunnies, right? They'll make more and more and more. All right, so let's take a look here. So this is an example of what I mean by these cells that have polarity to them. All right, and this is fairly common. This is an example of some cells here that have microvilli on the one end. So, or sometimes they can have cilia there as well. So we can have microvilli or cilia extending from these epithelial cells. And we can see here we've got the different kinds of junctions. So we've got the tight junctions where we're holding that surface together. I believe in the previous unit we looked at some examples of the tight junctions where in the lungs, the one side was held tightly together, and so the apical surface was held tightly together, but the base wasn't as much, was with the desmosome kind. And so that's kind of like the difference here. So you can see all the different kinds of junctions that might be holding the epithelial cells together. So you can see here there's a nerve ending, right? So when we say they're innervated, there's a nerve ending coming up here into the epithelial cells, but we don't see a capillary. We don't see that blood vessel coming in. And so these cells are avascular. And then down here is the basal region, that basement membrane. Okay, so it's the membrane on the bottom, all right, is attached to the connective tissue below. Right. So this is has polarity. One side of the cell is behaving in a different way than the other side of the cell. It might even have a different function. All right, so classifying. So we've got where, we've got what they do, function, we've got some characteristics of the tissue itself, and now we're going to start the classification part. So this is where you're going to want to pay attention so that when we're looking at pictures of tissues underneath the microscope, you know how to name them and classify them. I'm so sorry, my camera's being so silly. Okay, so epithelial tissue is classified based on how many layers of cells are there. So if it's one layer of epithelial cells, one layer of cells in that tissue, we say it's a simple epithelial tissue. If it's got multiple layers, like your skin, your epidermis, then we say it is stratified. Okay, so simple, one layer, stratified, like your epidermis here, many layers, multiple layers. We can also refer to the cell's shape, okay? So there are three basic shapes that we deal with here. Squamous, which are flattened cells, all right, like the cheek cells that we looked at when you were freshmen underneath the microscope. We scraped the inside of your cheek and we stained them that bluish color with methylene blue. Those are squamous cells, okay? The cuboidal cells, exactly what it says. They have like a little square shape to them. It's sometimes a rounded kind of square, but it's a cuboidal shape to them. And columnar means they're going like a rectangle. All right, so those are the two ways we're going to classify them. So, for example, you can have simple squamous cells, one layer, or you can have stratified squamous cells, which would be multiple layers. Right? You could have simple cuboidal cells, all right, or you could have stratified cuboidal cells where there's multiple layers of the cuboid cells, one on top of the other. You could have, again, simple columnar, right, or you could have stratified columnar. So when we start looking at examples, you're going to merge those two categories, how many layers and their basic shape, right? So something like this, that's what I just went through with you. Now, excuse me, for the columnar, we have this thing called pseudostratified. And These are kind of hard to identify, especially when you're trying to deal with the stratified columnar as well. So what is pseudo-stratified? Pseudo means false. So it appears that this is stratified or has multiple layers, but in reality, it does not. It's got one cell, and the position of the nuclei in that cell layer makes it appear to be stratified, but it's not. It's one layer, okay, and each cell, the nucleus, ends up in a slightly different position, making it appear stratified. So pseudo-stratified or false stratified, not really stratified. Okay. So here are some examples, and you're going to be learning how to identify these, and that's what your responsibility is going to be, actually, to be able to identify these different types of cells. So simple squamous, this would be a top view. So like if I was looking down on your skin this way, all right, that's simple squamous. Sorry about that. All right, stratified squamous. All right, actually this is probably not your skin. That's probably not the way to say that. That's probably the lining of one of your tracts there. This would be stratified squamous. So this would be like the layers of your epidermis here from the side. Okay, multiple layers of these flattened cells. Squamous is the flattened cell. All right. And then we have the simple cuboidal. All right, so again, we have a layer of cuboid cells, a layer of cuboid cells, a layer of cuboid cells. So just one layer, one layer, one layer, one layer around these little tracts. All right, stratified cuboidal, we have multiple layers. So there'd be a layer here, and then there's another layer here, and then there's one more out here. And then we're getting into the connective tissue by it. Okay. And our last one here, let's look. Oh, simple columnar. Sorry about that. So simple columnar, this one's nice because all of those nuclei are lined up nice in a row, and they're elongated cells. This would be stratified columnar. This is where things get a little tricky. All right, so here we've got a layer of cells here, and we've got a layer of cells underneath down here. Okay. This one almost looks like you've got a layer of columnar and a layer of cuboidal, but they're both columnar. Okay, this is a difficult one to identify. I personally think that's one of the hardest ones to identify when we're looking at them under the microscope. All right, pseudo-stratified columnar. What's nice about them is that they often have a microvilli or cilia attached, so that's helpful in determining whether it's a pseudo-stratified columnar or stratified columnar. But also they might often have a glandular component to them, so these little goblet cells here. And the nuclei just tend to be kind of random, and that sort of helps to do the pseudo-stratified. But honestly, I always kind of look for this fringe here, and that usually is my clue that it's the pseudo-stratified. And then we have this odd one that's called transitional. If I remember correctly, this is found in the urinary tract and in the bladder. So it looks like a mix of things, right? So it kind of starts sort of cuboidally down here, and then it sort of looks columnar, and then at the top it looks sort of flattened. So that's a little bit of a weird one. But again, the more practice you get with these, the easier they will get to identify. So that's it for the notes. And tomorrow we're going to start taking a look at these different kinds of epithelial tissue under the microscope and drawing some pictures and learning about where you would find the different kinds of epithelial tissue. So I hope that helps, and have a good rest of your day.